is our body in space, and I would suggest that these gentlemen have to have a pretty good sense of proprioception to carry out what this maneuver that they're doing. Each one of them has to have a very clear sense of where the other person is, as well as what forces are acting on individual parts of their body, and what kind of movements are going on at any point in time, or the whole thing collapses. A proprioceptor is the sensory receptor that allows us to bring this information into the central nervous system. Okay, and that's what we're going to focus on today, just like we talked about Meissner's corpuscles being sensory receptors. Today we're going to talk about proprioceptors, which are receptors in our muscles, actually, that allow us to determine this position. You don't have to learn any new pathways. Proprioceptors use the same pathway that our touch receptors do, that dorsal column medial lemniscus pathway. Again, I'm not going to ask you to draw it out, but that is the pathway they use to reach our level of consciousness. And the proprioceptors are innervated by the largest diameter nerves and the most heavily myelinated. Okay? We need to know very quickly and with very little, very low threshold at any point in time, where am I in space? What are my muscles doing? We don't want any delays in this pathway. Now, the one thing you have to keep in mind at all times, we're going to talk about what activates these proprioceptors, and it's mechanical forces. Similar to our somatosensory system, they were mechanoreceptors. We're going to see the same thing here. But if you think about it, at any point in time, are our muscles ever totally not contracted at some level of contraction? Even if you're sitting here in this room, you have muscle tone, right? So we're going to figure out, going through today's lecture, what is maintaining that tone even if I'm not moving? Now, I'm standing here, so I've got certain different forces acting on me maybe. But we're going to figure out in the next probably 2-3 lectures, what is it that maintains some level of muscle tone at all times, and what happens when that is disrupted and we do lose certain connections. Okay? So, big axons, heavily myelinated, and these are referred to generally as group 1 axons. So I'm going to be talking about 1A afferents today and 1B afferents, coming from slightly different structures. And those are the terminology that we use when we're talking about the innervation of these specialized receptors that are in our muscles. Little bit of terminology, and we're going to pick up on this terminology in the next, not the next lecture after this one, next Tuesday. There's this terminology called upper and lower motor neurons. It's not my favorite terminology. It's not a term I would have used, but again, nobody asks me, so I have to go with the flow and tell you what the standard is. And, you would look at this and you would say, okay, if it's up in the brain, we'll call that an upper motor neuron. If it's in the spinal cord, we'll call that a lower motor neuron. And that would be nice if you could do that, but you can't. Sorry. Okay? So, here's the definition that I want you to know. Remember how we started out with DRGs on the sensory side? This is terminology we really have to get for the motor side. When we say lower motor neuron, this is the neuron that goes directly out to innervate the muscle. Clearly, some of those motor neurons are in the ventral horn of the spinal cord. Could we have a lower motor neuron in our brainstem? You got a 50-50 chance, yes or no?
Yes.
Yes, okay. Now I could could get into more detail. But I won't, I'll tell you. Do I have cranial nerves that innervate muscles? Sure. Facial motor nerves, innervating my muscles of facial expression. Remember we talked about the facial nucleus was sending axons directly out to innervate those muscles. Neurons in the hypoglossal nucleus. Those go out to innervate our tongue muscles. Neurons in the cranial nerves that move our eyes around. The oculomotor, the abducens, the trochlear nuclei. Those neurons go right out to innervate the muscle. Therefore, by definition, doesn't matter that they're in the brainstem, those are lower motor neurons. Okay? We're going to go over this again, but any concerns or questions at this point? Okay? So if that's a lower motor neuron, so here's ventral horn cell, here's one of our neurons in the brainstem coming out of one of the cranial nerve nuclei, going out to innervate the muscle. Then by definition, an upper motor neuron, for the most part these are only in the brainstem. The cerebral cortex, they're in the brain, the cerebral cortex and the brainstem. But their job, so here's a neuron in the motor cortex, and you can see they're coming out and they're modulating the activity of the lower motor neuron. They do not go directly to the muscle. They either go directly to the motor neuron or they could use some interneurons. We'll get into those in an upcoming lecture. Those upper motor neurons could be in the motor cortex, or they could be in the brainstem. Remember that reticulospinal tract? I don't know, when we talk about the reticulospinal, we will be talking about it. But there are centers in the brainstem that give rise to input to neurons in the spinal cord. On Thursday, I'll talk about the vestibulospinal pathway. We'll talk about how the vestibular system modulates the activity of those neurons, okay? So lower motor neurons go out to the muscles, upper motor neurons modulate the activity of the lower motor neurons. So let's look at before we actually get into the proprioceptors, we need to do just a little bit of muscle anatomy and looking at how the muscle is actually innervated. This is not a course on muscle, so we're not going to get into great detail. So here's a micrograph of an alpha motor neuron, also known as a ventral horn cell, also known as a lower motor neuron. These are pretty big, about 40 microns. And those neurons, so here it is in the ventral horn, the axon leaves via the ventral root, comes out into your nerve, and it actually goes to the muscle fibers that are contracting. We call that the extrafusal muscle. So if you were to do a dissection and remove the skin and you were looking down right on the muscle, that's what we're talking about. The biceps, the triceps, quadriceps. Okay? So let's take a little piece of muscle out here and look at it. So skeletal muscle kind of looks a lot like when we, back in probably lecture 2, where we talked about what a peripheral nerve looked like. We said it was made up of thousands of axons. When we take a muscle and we cut it, what we see that the big muscle is actually made up of all of these individual muscle fibers. That's a muscle fiber, there's one, and each of these little circles is an individual muscle fiber, and every one of those muscle fibers has to get innervation. They have input from a ventral horn cell. That's one of the things we're going to talk about, how that happens. So I'm not going to go too much into any more of the muscle physiology, but these are basically multinucleated cells. There's our individual fibers. And again, each muscle fiber is going to get input from a single motor neuron. So, here's a definition I want you to know. Motor unit. And a motor unit is defined very simply as a single alpha motor neuron and all the muscle fibers that that motor neuron innervates. So again, here's our alpha motor neuron in the ventral horn of the spinal cord. Its axon comes out, gets to the muscle, and its axon terminates on a select group of those muscle fibers. In this case, they're the ones that are coded pink. Okay? So, each muscle fiber, and that is the only motor neuron that will innervate that particular muscle fiber. It makes sense. I mean, if that's a single cell, if I want that muscle fiber to contract, do I want half of it to contract? If it's upper motor neuron, if the motor neuron that's innervating the top part is active, and then the lower part of the muscle isn't doing anything, that doesn't make a whole lot of sense. It wouldn't be very efficient. So because they're these long cells, they get input from that individual motor neuron. But notice here, a motor neuron can innervate more than one muscle fiber. Okay, so you got that distinction? Muscle fiber gets one motor neuron, but a motor neuron can innervate many muscle fibers. So, I just like this image. This is a scanning electron micrograph. Here's that axon coming down and breaking up into its terminal branches. You can see here, this is synapsing. There's the terminal, what we call an end plate, making synaptic contact with that muscle fiber, and this one, and then it's kind of diving in, and it's going to pick up some muscle fibers that are out of the plane of this section. Say this is just the real thing compared to what I just showed you on the previous slide. Okay, motor unit size. So I said a motor neuron can innervate more than one muscle fiber. How many more? Well, that's variable. You can have large motor units. Here it's coming out, and this one is innervating 1, 2, 3, 4, say 5, 7, 8, 6 to 8 individual muscle fibers. Whereas this one, the blue cell, is only innervating 2 muscle fibers. Which one do you think would give you the greatest control? A large motor unit or a small motor unit? Small. I kind of heard it. The small. That means that motor, kind of like what we were talking about, receptive fields, the very tiny receptive fields allow for much more precise localization. A small motor unit is going to allow you to have much finer control over the contraction of the muscle. Where do you think you might find your smallest motor units?
Ears.
Ears? Oh, fingers. Fingers, yeah. Yeah, you've got some muscles in your ear that control the little ossicles. Yeah, they'd be pretty small. How about our eye movements? Okay. Especially here in finger movements for control. I don't necessarily want to have to grasp everything. I might want to very slowly and precisely wrap my hand around something. So, Small motor units are going to provide that very precise control, and large motor units, you'd find probably more like your back muscles. I just need my back muscles to contract to keep me upright. Even your biceps, those are going to be pretty large. I want that muscle to contract when I'm picking something up or bending. Okay. Now, here's another term I want you to know. Motor pools. And here's our definition. This is a group of motor neurons here in the ventral horn of the spinal cord whose axons all innervate the same muscle. Okay? So remember I showed you that picture. Well, I'll show you it on another image. We'll look at that again. So here are these kind of pools of motor neurons. And they have a precise, again, here's that term, somatotopic organization. We talked about the somatotopic organization in the sensory and the motor cortex, or in the sensory cortex. We haven't done motor cortex yet. And in the spinal cord, the neurons that are located medially innervate our trunk and our proximal muscles, shoulders, arm. Motor neurons that are located laterally innervate more distal muscles, like our hand, our fingers. Same thing for the lower leg. Toes are going to be out more laterally, and as you move in, you're going to get leg and thigh. Okay? Remember, this is a somatotopic organization. We are going to come back to that. So, are you okay with differentiating a motor unit from a motor pool? We're going to see why this is significant. So, these motor pools, again, all the neurons that innervate a single muscle, not a single muscle fiber. That would be important on an exam. If I were to ask you to define that. Okay? These motor neurons, these pools, tend to be organized in these columns. What that means is that you could have more than one level of the spinal cord going out and innervating a given muscle. Like the biceps gets innervation from, I can't remember it now, C4 and C5 and 6, say. And you can see here, here's this purple neurons in column B. Their axons come out, and as they merge, they actually do come together usually to form a common nerve. And the purple ones are coming over here to innervate muscle B. Here in the green column, we see some coming from L5. We see this one coming, the red one coming from L4, but it's in the same column. And those axons are coming out to innervate muscle A. So we're beginning to see this precise organization in this pattern of innervation. Okay, so right now, like I said, your muscles are at some state of contraction. But what if all of a sudden you decided you wanted to pick up your water bottle? Would you have to increase the muscle contraction, say of your biceps? Would I have to increase the contraction of my hand muscles to get hold of that bottle? So I've got to have some mechanism whereby I can increase or decrease the strength of the contraction of a muscle. You know, am I lifting weights today, or am I just lifting my coffee cup? Okay? So let's look at the role of the motor unit and the motor pool. We're going to use a term called recruitment. So motor unit, okay, so I got one nerve, I got one neuron. So what can I do that might increase the strength of the contraction of its subset of muscle fibers? Okay, so the answer is we can change the firing frequency of that motor neuron. So here's my motor neuron, and I'm going to stimulate it. That's what this little curly Q thing is here. That's a stimulation. And I'm going to let it generate a single action potential. Action potential is going to travel down, and it's going to reach these muscle fibers, release the neurotransmitter, and the muscle fibers are going to contract. Okay? So, one action potential, I get what's called a twitch. Okay, that's not too impressive. But what if, so here we stimulated, we got a little twitch, we stimulate again, I get a little twitch. What if I increase the frequency of stimulation? You guys remember from 3000 how to summate an excitatory or an inhibitory postsynaptic potential? You thought you'd never have to think about it again, right? But I know what Dr. Boyd tells you in 3000. Okay? And you do it by summation. You bring your stimuli in so that they're occurring closer together. So here the stimuli were separated so that we had the twitch and the muscle returned to baseline, and then it twitched and then came back to baseline. But what if I start increasing the firing rate? So now it doesn't get all the way back down, so when we get the next stimulus, it begins at a higher point. So now we get an even stronger twitch of the muscle. And now, again, it's starting at an even higher point. And we can keep doing that until we reach a point where we have what's called tetanus. And tetanus basically means kind of the maximal force that you can generate. That muscle is totally contracted, as long as that stimulation is present. So at the highest rate, we're in complete tetanus. Here's a couple more definitions. Tetany is a sustained muscle contraction due to the stimuli being repeated so rapidly the muscle can never recover. And it's all due to temporal summation. I hate to have you go back to classes you had a couple years ago, but so this is the motor unit. This is what the motor unit can do. So now instead of having just small little contractions, I can make that muscle contract and hold that contraction as long as I'm giving it inputs, which might be trying to pick up that weight. As long as I have that 5-pound weight in my hand, I'm activating that. Okay, but again, this is at the level of individual muscle fibers. What would be another way I might be able to make that muscle have even more force? I know you're all still back on the beaches. What about the motor pool? What if instead of activating, oh, whoops, I forgot. Did you ever wonder where the term, you all got tetanus shots, right? At some point in your life, and you'll continue to get them every 10 years. And this is kind of where this term came from. These are little Clostridium tetani, little spores that live in the dirt. So you get a cut, and they have a really interesting cycle. You don't have to know, this is a freebie. These spores can bind, if you get a cut, you get exposure of any of those peripheral nerves, they'll actually bind to that peripheral nerve terminal. I don't know why, axon terminals like to pick things up in their environment. And they'll pick up the spore and they'll transport it retrogradely back toward the central nervous system. And then it will go and it will bind to proteins, specifically at inhibitory endings. And what they do when they get into those endings is they block the release of GABA. That means there is no inhibition, and once the motor neurons are firing, there's nothing to shut them down, and this is kind of the classic image of somebody who's in full tetanic rigor. All of your muscles are in full contraction because there's no inhibition. We need inhibition to shut things down. So this is why you want to get your tetanus shots, because you don't want to wind up in this position. They're nasty little critters. Okay, you don't have to know any of that. I won't ask you about the tetanus bacteria. But let's get back to how can I get my muscle to contract more? And this is now, let's recruit more than one motor unit. Let's get more of the neurons in the motor pool to do the same thing. So, come to the next slide, this makes more sense. So here's my motor pool, and this little pink one is innervating these muscle fibers. This kind of salmon-colored one has its population of muscle fibers, and the beige one innervates its muscle fibers. So if I only activate motor unit 1, I'm only going to have these lighter colored muscle fibers contracting. But what if I activated all of them at the same time? I could get all of the muscle fibers to contract. So one way I can get a stronger contraction is to activate more of the motor neurons. So what is the corollary of that is that means that at any given time, we are not activating every motor neuron in the motor pool. Unless you're really doing something with tremendous force. You know, if you're trying to lift a 500-pound weight, you might be trying to activate all of those motor neurons, and you may or may not be able to lift that weight. Okay, questions? So these are kind of important concepts that we're going to circle back to these at the end of the lecture today. So we have some idea of how we can make our muscles contract. But that still doesn't tell me what the proprioceptors are doing. What's their role in all of this? So finally, we're going to get to that. So there are basically two kinds of proprioceptors that we're going to talk about. One is called a muscle spindle. So here's the big extrafusal muscle fibers, and embedded within those extrafusal muscle fibers is this structure, and I'll give you some more definition of that in a minute, which is attached to these other muscles. So if that muscle contracts, the spindle is going to change shape. If the muscle is stretched, the spindle is going to change shape. It's kind of in parallel with your muscle fibers. The other proprioceptor we'll talk about is called a Golgi tendon organ. And as its name implies, it's not embedded in the muscle fibers, but it's related to what's going on at the level of our tendons. So let's look at the spindle first. So what are they good for? They can detect change in muscle length, position, how quickly is a muscle contracting, at what rate is it contracting. So they're getting a lot of feedback on the state of contraction of these muscles. So again, here's our big extrafusal muscle fibers. Well, here's an actual histological image. There's your extrafusal muscle fibers, and embedded within that is this specialized little structure called the muscle spindle. And again, it's in parallel, and it's actually attached to those fibers. So what do they consist of? These are actually mini muscles. So just like we had, I showed you that big image of the big muscle with individual muscle fibers, the spindle also has some muscle fibers. We call these intrafusal. Extrafusal are the outside ones, intrafusal are the muscle fibers related to the spindle. And there's a big area, just like we saw in the extrafusal muscle fiber, where there's a bunch of nuclei collected, the nuclear area, and all of that is surrounded by this capsule that attaches on either end. The muscle spindle is innervated by a type 1 or a group 1 muscle nerve. Largest diameter, heavily myelinated. So let's take all the extrafusal muscle away and just look at the spindle itself. So, first of all, there is striate muscle in there. Although we don't have much voluntary control over it. And but what their role is to determine how tense the spindle is. And again, we'll come to why that's important. So it is very sensitive to changes in the length of the muscle, and in particular, to stretching of the muscle. Now, in and of itself, that wouldn't get many signals back to the CNS. So remember those 1A afferents, that type 1, these are called 1A because they're the largest diameter. That axon, the cell body of origin is in the dorsal root ganglion, because this is a sensory axon. So here's our crossover, where we're talking about both motor and sensory at the same time. Cell body is in the DRG, peripheral process comes out, enters the capsule, and literally forms this spiral around the nuclear region. Okay? When that spindle is stretched, that opens channels on the endings of these axons. Remember when we touched, we said they were mechanoreceptors, and that pressure would open channels, allowing influx of sodium. Sufficient sodium flows in, generates an action potential. The action potential goes back toward the cell body, just like we saw in the somatosensory system. There are some, what we call secondary endings, and they just kind of go to, they're not right around the nuclear region. We're really not going to focus on the secondary ending. Again, if I was doing a muscle physiology course, we would be doing that. But I'll at least point them out. And then, see these little red axons coming out that are going to your intrafusal muscle? Those are called gamma motor axons. Gamma motor neurons are in the spinal cord. We're going to put this all together. But right now, we're just looking, here's the components. Got intrafusal muscle fibers. We've got the primary 1A afferents. And we've got, for our purposes, we've got the gamma motor neurons. fat and the other one is kind of skinny, and that's true. They're called nuclear bag, nuclear chain fibers. They do have some different physiological properties. We're not going to get into that, okay? We're just going to talk about these afferents coming out to surround the nuclear area. If you've taken the muscle physiology course, you know more about it probably than I do. Okay, so here's our primary afferent. Oops, let me back that up. So here's our 1A afferent. Comes down and it forms that loop around the nuclear region, and that's usually referred to as the annulospiral ending. So that's the terminal process. Here's the important thing. Those channels open when the muscle is stretched. Now, I don't have to stretch my arm out completely to activate those channels. Very, very micron changes in tension are sufficient to open a channel on that ending. I'm sure every one of you has had your knee tapped in a doctor's office. Does he have to whack your knee very hard? And what happens? Your muscle contracts, right? You kick out. So he doesn't have to use it. That is sufficient stretch. Just that hammer tapping is literally stretching out that muscle a little bit to cause it to, and we'll go through the contraction pathway here in a minute. But it is important to know these afferents are activated by stretching the muscle, not by contracting. Okay, and we have the type 2 that are out at the end there. Okay, the 1A and the type 2, again, are sending information back to the CNS. And again, here's our little gamma motor neuron. Always look at the direction of these arrows. That's telling you which way the action potential is moving. This action potential is coming out, and it's innervating that intrafusal muscle. So let's put this together, see what's happening. Here's our DRG cell. Here's our ventral horn cell in the spinal cord going out to innervate the muscle. That's supposed to be the muscle spindle. That's supposed to be the little annulospiral ending. If I stretch the muscle, that causes information to come back toward the DRG cell, enters the dorsal root, comes into the spinal cord, goes right on through, and goes directly to the alpha motor neuron that innervates that muscle, or the motor pool or a motor unit, depending on how much stretch is applied. And that then goes out and causes your muscle to contract. So at all times, our muscles are under some tension. Whether we got gravity acting on us, even if you're sitting in your chair, you don't realize it, but you're making very small little movements. It's causing activation of those 1A afferents, which are then activating probably not a whole lot of motor units in this case, just enough to keep some tone. Okay, let's look at it here. So here's our extrafusal muscle. Here's our muscle spindle. And if I stretch the muscle, I'm going to see action potentials in the 1A afferent. When that happens, the muscle contracts. So something that's suspended between 2 points, and I stretch it, open the action, open the gate, ions come in, we generate an action potential. What's going to happen when the muscle contracts? Is my muscle spindle going to stay stretched? Now, as the muscle contracts, as you can see on the image here, the spindle relaxes. You don't just put a piece of string between your fingers if you want to play with that later. Okay? Well, that's a little bit of a problem. If my muscle contracts, I no longer am opening channels. If I'm not opening channels, there's no signal going back, and therefore, my alpha motor neuron's not going to be firing, and I'm not going to be getting contraction. Do I want my muscles to just go limp? They contract once, and then I just kind of turn into jelly? Not very attractive. So, we've got to come up with a mechanism now that prevents that from happening. And that's our friend, the gamma motor neuron. Okay? So, these gamma motor neurons are also in the ventral horn of the spinal cord. But remember, they're coming out to innervate the intrafusal. There are these green axons that are coming out here. They're innervating the intrafusal muscles. So let's do my diagram here. Dorsal root ganglion, alpha motor neuron, gamma motor neuron, muscle with the muscle spindle. Let's stretch the muscle. I'm going to activate that spindle. Action potential is going to come back. High little DRG cell, see you later, goes into the cord. Synapses on our alpha motor neuron, which goes out and makes the muscle contract. So putting those last couple of slides together. And now our spindle is no longer functioning. But remember, the ends of the spindle are muscle, which means it's contractile. So what if I made that muscle contract? Would that replace the tension? What if I activated my gamma motor neuron and it came out, and now here the muscle fibers are still contracted, but now I've also made my intrafusal muscles contract. Does that restore tension? The answer is yes. And you can see that again, if I'm recording from that axon, I'm again generating action potentials. Now, it may not be as strong as it was when we just had the initial stretch, but it's enough to continuously provide some input to our alpha motor neurons to maintain some level of contraction in the muscle. Does that make sense? It probably doesn't right now. You have to think about, you have to go back and look at it. Let me give it to you a different way. You might say, well, what's making the gamma motor neuron fire? And what's making the gamma motor neuron fire are areas from our motor cortex and from our brainstem. Our vestibular system in particular is very sensitive to perturbations in movement. It's going to tell those gamma motor neurons, hey, we need a little more tension in these back muscles, need a little more tension, motor cortex. I'm trying to pick something up, maybe you can get a little more tension there.
So will this all compound? Like as the muscle contracts, will the spindle then contract more, contracts? Or was this just a one-time thing with the gamma motor neurons?
Well, it will be sustained in terms of there will be continual opening of those channels and continual level of contraction in our muscle. Now, we will come back to, but what if I need more power? Okay? So right now, I'm just assuming you guys are all just sitting there, you're not, can't really use a whole lot of it. So maybe I'm only activating, 20% of my muscle fibers. And those will stay maintained at enough tension. Okay, if you don't like my diagram, and if you use this slide, do it in slideshow mode. Otherwise, it won't make any sense. This is referred to as alpha-gamma co-activation. So our 1A afferents and descending information, like if my motor cortex wants me to voluntarily move, tells the alpha motor neuron to send an action potential out to the muscle. Causes contraction. That leads to decreased tension on the muscle spindle, decrease in 1A afferents, and theoretically your muscle would relax. Every time you contracted, everything would relax. Then you have to contract again and relax. Okay? But at the same time, especially these descending afferents, go to the gamma motor neurons. They contract the intrafusal muscle. This is why you have to do it in slideshow mode. That maintains tension on the spindle. That maintains activation of 1A afferents and maintains some level of activity in our alpha motor neurons. Okay, think about it. You can ask me questions on Thursday if you're not clear. So, I'm going to summarize our muscle spindle. There are 2 efferents. In yellow, these are the axons of the alpha motor neuron going to make our muscle contract. And in green are the axons of our gamma motor neuron, which are causing the intrafusal fibers to contract. We have afferents in blue here, wrapping around the nuclear region that are detecting when the muscle is stretched, and that signal is then carried back to the spinal cord. When I was a physical therapist, we used a technique, which now at the time I was just a term, called proprioceptive neuromuscular facilitation, PNF. And this was used in patients who were paralyzed and we were trying to get some return of function in those muscles, trying to get them to contract. And that was exactly the technique. They would maybe not be moving it, and we would take it and just gently put some pressure on it. And you could get that muscle to contract. And we're going to talk about why that happens, even if you have a stroke. And over time, that would build up, and hopefully we can be maintained and then build up some of the muscle strength and some new pathways to keep the muscles moving. Now I would know what we were doing at the time. Okay, very briefly, the other proprioceptor is called a Golgi tendon organ. And as their name implies, these are also afferents, but they don't go to the spindle. They intertwine between the collagen fibers of your tendon. Tendons are not contractile. They're just a bundle of connective tissue, and that's why they get damaged easily, get torn, get stressed, because they don't tolerate a whole lot of tension on them. These are just slightly smaller, so they're called 1B afferents. And their job is kind of just the opposite of the muscle spindle. Think about it, your tendon is attaching to the bone, and it's attaching to the muscle. So if a muscle is contracting, and that tendon can't contract, or has very limited flexibility, as the muscle contracts, it's really putting tremendous tension on that tendon, right? The job of the Golgi tendon organ is to basically shut down the system. If you're overexerting to the point where you could damage that tendon, these guys come into play and they basically inhibit the motor neuron that's going to that muscle. So, in this case, the Golgi tendon organs are active when the muscle contracts. The spindle was active when the muscle was stretched. So, here's the muscle at rest. Here's our little Golgi, the axons coming in, cell bodies in the DRG, no activity, the muscle's resting basically, no activity. But now the muscle is contracting. So the tendon is getting pulled one way by the muscle and it's attached to the bone. So it's kind of like getting pulled in both directions. And then notice we start seeing activity as tension increases. So what's happening here? So here's our flexor muscle. Here's our ending of the Golgi tendon organ. If that muscle starts contracting, here's our excitatory neuron going to our extrafusion, oops, which one do I want? I want the red one. Here's our neuron going to the biceps muscle that was contracting. The 1B afferent comes into the spinal cord and it projects to what's called a 1B inhibitory interneuron. So, the first one I showed you was monosynaptic. 1A comes in, goes to the ventral horn cell goes out. Now we've got another little interneuron that's sitting in the spinal cord, and it inhibits the motor neuron that was going to the contracting muscle. It will also project to an excitatory interneuron that activates the muscle on the other side of the joint. So if I'm flexing and I'm putting tension on my biceps tendon, this shuts down the biceps and activates the triceps. Now, it's not all or none. It doesn't totally shut down the biceps. It doesn't totally activate the triceps. But it says the triceps are going to kind of try to counteract that to, again, prevent you from doing damage to that tendon. You always hear about people, trying to lift something, weightlifters at the Olympics, trying to lift that 700-pound barbell and snap a tendon. Not a good thing. So, this information, I said it uses the dorsal column medial lemniscus pathway, gets to VPL thalamus, and in this case, that proprioceptive information goes to area 3A. Remember, our postcentral gyrus is the primary sensory cortex. We divide it into area 1, 2, and 3. Cutaneous receptors go to 3B, and the proprioceptors just a little bit deeper in that central sulcus. But they also, just like 3B, these also are projecting to area 1, area 2, association areas. So remember, So as we go from area 3, progressively more caudal areas of this field, we get area 3 is going to be very localized. Area 1 maybe gets convergence of information from multiple digits. Area 2 even more. Well, now add on to that, it also gets information not only on where something is touching, but where our fingers are in space. And remember our little monkey trying to get the grape out of the glass? Well, that's where the proprioceptors also come in. Because if he has no clue where his fingers are, there's no way, not only can't he feel properly, but he's not able to use his sense of proprioception. Okay. Last thing we want to talk about are reflexes. There are circuits in the spinal cord that are, what we've been basically talking about is a series of spinal reflexes. Somebody taps your quadriceps tendon, your knee moves out. That signal did not have to get up to my level of consciousness. That just happened at the level of the spinal cord. I activated the 1As, the 1As made the muscle contract. Patients with spinal cord injuries have reflexes. They are not totally, they don't lose that muscle tone. In fact, it gets even higher, which we'll talk about later. So, a reflex at a minimum needs a sensory input and a motor output. Most reflexes are a little more complicated, as we saw with the Golgi tendon organ. Sometimes there are interneurons that are also involved. But what we have been describing is a simple input-output pathway. And that is going to function regardless of whether there is input coming down from higher levels. Okay, so pretty much what we just said here. There's a discharge, there's an afferent input, there's a motor output. And again, even if we cut the spinal cord below the level of the lesion, you would still be able to elicit a reflex by tapping the knee. The supraspinal control is important for when we want to have directed movement, not just reflexes. I want my hand to do something, so I'm going to specifically activate a population that's going to allow that to happen. And a lot of the other role for these descending inputs is to control the level of excitability of these reflexes. And they do that by using a lot of those inhibitory neurons. So if I lose my descending control, those reflexes are just free running. And they're just going to be pretty much doing what they want to do. So, we've been talking about a monosynaptic stretch or extensor reflex. We talk about patellar, but you can do it at your ankle. Anybody had their ankle tapped? The tendon on the back of their ankle tapped, and your foot should pop down a little bit. You can do it at your biceps and get a little jerk of your bicep, your triceps. You can do this anywhere where you have access to the tendon. Now, we can make this one more complicated. Why do we want to do that? Why not? You're back from spring break. Okay? So, here's our 1A afferent. Activates the alpha motor neuron that goes to our quadriceps. Our quadriceps contracts. Basic stretch reflex. But at the same time, it probably would be a good idea if we shut down the activity of our hamstrings a little bit so that they're not trying to fight against us. So you could have a stretch reflex with what's called reciprocal inhibition. And again, here's our little interneuron, and it's going to inhibit the alpha motor neuron that goes to the muscles on the other side of the joint. Again, it's not all or none. It's just kind of that nice balance so that we can control it. So here's a practical example of the stretch reflex. So I'm holding a mug. This person is holding a mug. And there's a certain level of activity in the 1A afferent coming out of the biceps muscle, causing that muscle to contract. There's probably some inhibition of the triceps. And then a beverage is added to your mug. This is rated G, TG, just some soda. You can put whatever beverage you want in there. Okay? So now what's happening is, I'm holding the mug, but it's now heavier. That's putting stretch on your spindles. So now, we have to go back to that idea of recruitment so that I don't spill that all over the floor. So how could I prevent that from happening? Well, as I'm stretching, I'm probably increasing activity in that 1A afferent from what it was at rest. So that means the firing rate of individual motor units is increasing, putting more tension on their respective fibers. And at the same time, I may be activating more neurons in the motor pool that go to the biceps. So I have two ways of compensating. Increase firing frequency of a motor unit, increase the number of neurons that are active. And I can then bring it back up to a nice level position. So there's a practical example of recruitment. Okay, last reflex. Withdrawal reflex. Pain. You step on a tap, you burn your hand. There's something sharp. What's your natural inclination? Pull it away. And there are special afferents in the DRG, special neurons in the DRG, again, pain sensing. This takes us back to our pain fibers. There are pain fibers in your muscles, in your skin, obviously. Not in your muscles necessarily for this, but in your skin. And I'll show it to you here first and then on another diagram. So, I touch something hot. That's going to go to a pool of excitatory, this one again, it involves, the pain pathway just for whatever reason just seems always more complex. But it involves a lot of interneurons. First thing we're going to do is get an excitatory interneuron. They're going to then excite the alpha motor neuron. And that's going to go to our flexor muscle, whether it's in the leg, in the arm, wherever. And we'll get contraction and withdrawal from the pain. Make sense? Even though there's a bunch in there. And we may also activate inhibitory interneurons that again go to the alpha motor neurons on the other side of the joint, which are usually called the antagonist muscle. Okay, so that's basically the same pattern of what we're seeing. It's just that they're being activated by these pain-sensitive axons to get us away from a situation. Okay, let's add one last level of complexity. Especially if it's in your lower lip. So here's somebody stepping on a tap. Okay? And these are, again, our nociceptors, probably the A deltas. That sends that signal into the spinal cord. Here's our interneuron pool that's going to excite our flexor muscles, our hamstrings. So I'm going to pull my foot back, hamstrings flex. And I'm going to inhibit the extensor muscles. Here's an inhibitory interneuron, so I'm not continuing to put pressure on that foot on the tap. Now you think about it with your foot. If I flex my right foot What should I be doing with my left foot? Hmm?
Pushing it down, thanks.
Yeah, you want to be extending it, right? Or you could fall. You could just fall over
No.
in pain. You could do that too. But most of us don't want to do that. So I'm going to flex on one side, but I need to extend on the other. So this is flexor withdrawal. Now, here's a group of interneurons. They cross the midline, and they do just the opposite. They're going to inhibit the flexor and excite the extensor on the contralateral side. If you think about it, even if you don't look at the diagram, it just makes sense as to what you want to do. I want to flex on one side, I want to extend on the other side, and most of that is all occurring at the level of the spinal cord itself through these interneuron pools. With excitation for flexion, excitation of extension on the other side. Now, probably if it's something with our arm, we're not invoking that, or, I don't usually do this. But you can, and again, when you're treating patients, that sometimes is one way to elicit some of that movement, is to try to extend something out, or to flex on one side to get extension on the other side. Had I known then what I know now, it would have made more sense. We just did it, because it worked, kind of. And that's why I went back to grad school, because we were doing these things and I just didn't understand what we were doing, why they were working. So that's why I wound up here. Okay, so that's just in words what we just talked about. So I put a summary in here about what we just talked about.